Okay, perfect. Yeah.
Some feedback.
Yes. David, can you hear me? Oh, thumbs up. Okay, so I'll give you a quick introduction, David, and then we'll ask you to unmute. Does that sound okay? Perfect, great. Okay, so it's my kind of great pleasure to introduce our next lecturer, David Norris. David has been a pioneer of laminar fMRI for many years, both in terms of development of technologies, applications to cognitive neuroscience, has sort of a unique perspective on the field, having been involved since really the beginning. And so we asked David to give us a lecture on applications of laminar fMRI, including history and lessons learned. And so David, we're really happy to have you here. I'm going to go ahead and put your video, if you don't mind. Well, people are on Zoom. I'm going to put your video on the screen. If I can. Oh, very good. And just off in the corner. And so, David, when you're ready, you can unmute, and we're ready for your lecture. Oh, no. And then.
Okay, am I audible?
So we don't.
Am I audible
So
now?
we can't hear him in the room?
Test my volume.
Oh, yes, that's perfect.
Okay, all right.
Too loud? A little bit too loud? Try one more time, David. Just a little bit too loud.
Yes, well, I was maybe shouting a bit then. This is about the volume I'll speak at.
I think that's pretty good. Is that too loud?
Okay.
Great.
Yeah.
Awesome. Thanks, David.
Okay. Thank you, John, for the kind invitation, and I'm sorry I can't be with you in person, but I see the faculty, and I think you're going to have a great course. I've been given this title by John, which I really quite like. History, Key Applications and Lessons Learned. I think this is kind of teasing me to all sorts of statements. So I'll give you a history of laminar fMRI from a fairly personal perspective, but I will try and include major developments. Okay, so we'll start off with a short introduction, and I'll then show you some historical insights. I'll then move on a bit to connectivity and the special role that laminar fMRI can potentially play in connectivity. And I'll kind of follow the Carl Friston doctrine of splitting connectivity into functional and effective connectivity, and give two examples at the end of studies which show some measure of effective connectivity in the language and the visual systems. And then I will draw some general conclusions. I'll also try to draw some sort of intermediary conclusions after each section. If I'm getting dramatically behind time, I will probably leave out one of the effective connectivity studies. And if you want to ask a question, please, well, there will be time for questions at the end, but also if it's a burning question, I'm sure I can find a way of answering it. So why do we do this stuff at all? And I think the simplest answer, and the one you most commonly encounter, is to look at laminar response profiles, which are interesting. And if we're fortunate, we may even be able to see on some occasions the role of a specific layer in a task. But we can also hope ultimately to examine the interaction between layers, and that can be within a single region or with another region. And I think people even in the 1990s were realizing that this kind of study had a great potential for fMRI. And a lot of this stuff started off in rodents, and I know you've had a kind of anatomical lecture for John, so I'm not going to repeat it. But what I'm particularly interested in the first part of this lecture is the vascular structure. And if you look at the vasculature, we have a mass of vasculature on the peel surface, and then we have descending arteries and veins. So in terms of our fMRI experiments, unlike a normal fMRI situation, we have to contest with blood flow from the deeper to the superficial layers. And this will be a recurring theme, I suspect, throughout the week. And just for your information, there are, of course, the 6 histological layers, but there are also different levels of vascularity as a function of depth, which don't strictly match with these. So if we go back now to the turn of the millennium, then some people from CMRR were already looking at this kind of stuff in rodents. And what you see already is looking at the bold response in the rat, looking at the CBF response in the rat, getting a laminar response where if you look at the signal change in percentages, again, the bold signal change is building up towards the surface, where this isn't the case with the CBF. You can also notice with the correlation coefficient, it's not so bad with the bold, but most people look at bold in terms of the size of the response as a function of the depth. If we move on a few years to some experiments done by Noam Harel in the cat, people started to wonder, of course, what would be a good contrast for doing laminar fMRI, because the gradient echo signal is obviously accumulating on the surface. The spin echo is looking somewhat better. You would originally expect in this kind of experiment to have an increased activation somewhere deeper in the cortex, around layer 4 for the input layer. And if you look at CBV done with Mayan, then this is a kind of gold standard-y experiment where you can see indeed that you have most of the activation in the cortex. The only thing is, of course, nobody wants to have Mayan injected into them. So we move on a few years further, and we see from Chunji Kim's lab examinations of spin echo with low and high threshold here, and Mayan again showing the gold standard. But now we make the important step towards vaso, which is a non-invasive way of measuring CBV changes, and showing that essentially in the right situation there is a bridge between Mayan and vaso. So we could hope, for example, with a vaso-based technique in humans also to get a truer profile of activation. So this is the kind of, in terms of human MRI, this is the kind of prehistory that we had. And then getting up to about 2010, the activity somewhat increased in humans. And we had to work out where to go. And we started off looking at the animal experiments. Well, we have a clear hierarchy of specificity. CBV and CBF look to be the best. Then we've got spin echo bold, and probably gradient echo bold comes in last. But if you want convenience and sensitivity, probably the first thing you're going to look at is the gradient echo. And I couldn't possibly let a talk at MGH go by without showing John's famous slide from his 2010 publication, where I don't know if it's the M from MGH or the M from Martinos, which is being activated, but this is an imposed pattern of activation on human visual cortex. And you can see that you have better specificity down at the lower depths and better sensitivity towards the surface, but the specificity is suffering. And around the same time, we published a study at 3 Tesla, looking at activation in human V1 and showing a very similar type of activation pattern where you see most of the activation that you're recording in a normal fMRI experiment is coming from out on the peel surface. But we actually co-registered for stripe of Gennari, and then you do see a slight bump here, but this is not what you would normally be measuring. We also tried at 7 Tesla, and this was just a single subject measurement at 7 Tesla with gradient echo. And if you see here the anatomical image, the T2 star weighted image, the activation map, and then you zoom in on them, calculi and sulcus, with the stripe of Gennari in the blue arrows. And actually in this particular subject who was exceptional, you could actually see the activation again in the stripe of Gennari. So if you do have some extra anatomical knowledge, you can match your activation more, but generally speaking, you're going to get activation which builds up towards the surface. So the early experiments with gradient echo showed that there was some promise of doing laminar fMRI, but also that we have a heavy bias towards the peel surface. Of course, we were doing 3 Tesla because we were rather cautious, and then the question arose with us, well, what do laminar profiles look like at different field strengths? And actually, back then we had a 1.5 Tesla, a 3 Tesla, and a 7 Tesla. And if we just look at straight activation profiles, you can see obviously the 7 Tesla is doing better, but the qualitative nature of the profiles is not changing very much between the field strengths. And even if you would take the ratio of the parenchymal signal to the peel surface, it doesn't change very much. So the question is, of course, why is this? We all know 7 Tesla is vastly more sensitive than 3 Tesla, etc. Well, the answer is, if you're pooling your data to create profiles, actually you fairly rapidly reach the physiological noise limit, and that's the point where the physiological noise is dominant, and further averaging, so to speak, adding more voxels in will not increase your SNR. So for just these very simple measurements of activation profiles, the field strength, there is an advantage for 7 Tesla, but it's not absolutely decisive. However, of course, we would want to be more ambitious than this, maybe, and I'm not sure always that the laminar profiles are the most exciting things we can measure. On the other hand, we do now see a movement encouraged also by Renzo, who's been leading a lot of the stuff on vaso. We now do see a movement towards going to 3 Tesla as well, just to broaden the scope and the availability of laminar fMRI. So we actually found you reach the physiological noise limit pretty quickly for all field strengths. But of course, when you go to small patches, you'll get more sensitivity at higher field strength. And this is about looking at the main effect of task. And as we go further into this talk, you will see I get less and less interested in the main effect of the task. So that's where we got to with field strength, where we got to with gradient echo. And we should, of course, celebrate our failures a little bit. And this is where we reached the limit with the gradient echo. We had the smart idea of doing black and white stimuli versus color, and from theory, we should expect more color processing in the superficial layers in early visual areas. But we then had the confound that we also got more activation with the color stimulus. And try as we might, we could not really prove that there was a higher level of activation in the superficial layers. And this is the sort of problem you can encounter if you use gradient echo, looking at some relatively naive differential paradigm. It gets quite challenging to actually interpret differences, because you've got the carryover of signal from the lower layers. So you could try to do something about this. And the obvious thing to do is to try to simulate your point spread function. And we did this some years ago, and there are now more sophisticated models. But basically, if you look at a spin echo and activate a specific layer, you will get a sharp activation with no build up towards the surface. Whereas with gradient echo, you get the strong activation plus a flat, pretty near to flat tail going to the surface. And of course, it's the accumulation of all these flat tails that leads to the increase in signal towards the surface when you're activating. So can we deconvolve these? Well, we had the multi-echo data, so you can see how you have a different profile going from white matter to CSF as a function of depth, but you know that the underlying activation was the same. So if you deconvolve, you should get something fairly similar, except of course the strength of the activation is different. And then if you normalize that, you get a much closer clustering, except of course at towards the peel surface. So this is an area, if we're drawing another conclusion, this is an area where more sophisticated models are available and coming along, which can also deal with the time variance in the signal, and where it may be possible to correct for gradient echo signal bias in the future. So after this prelude of some methods aspects and some acquisition aspects, maybe we can look at some examples from human neuroscience which have made a difference. And I'm not going to give a comprehensive overview because there are quite a lot of them now. One of the early ones we went to when we started talking to our neuroscience colleagues was, could we get some kind of surprise effect in human V1? So one of the attractions of doing laminar fMRI is ultimately that we may be able to test theories of how the brain works, like predictive coding, where you have top-down predictions and they mismatch the bottom-up input. And if you present a shape like this, then you cannot avoid seeing a triangle here. But if you would rotate one of these Pac-Man shapes, the triangle illusion would disappear. So we get some excitation by prediction because there's no real input here, but you think you're seeing a triangle. And we have an inhibition of signal around here where the Pac-Man are. So this was work done in the group of Flores de Lange, and they'd already shown that if you did normal fMRI, you would have a signal. If you look at the condition illusion minus non-illusion, then you would have an increased signal where the triangle was and a decreased signal where the Pac-Man were. Right. So we then basically took this experiment and laminarized it, if you can use such a term. So we then get laminar profiles of activation with or without the illusion. And the point is, when we have the illusion as compared to not having the illusion, what starts leaping out at you is actually a signal difference in the deeper layer, which is highly significant compared to just flashing a checkerboard in somebody's eyes and getting a pretty normal profile. So this was very encouraging for us, and I think many people in the community, that you could do this kind of experiment and get an interpretable result out of it. Just for completeness, there's also the reverse situation when we're looking at the Pacman objects, but indeed there's a small difference here, but it's not as dramatic as the signal difference you're seeing in the deep layer for the illusion. Again, when you've got the checkerboard, you get the standard increase in signal towards the surface. But of course, this was an age in which laminar fMRI was being done primarily in the visual or in the motor regions. So I'll jump in for another application to actually a famous paper which Emily Finn and Renzo Huber published in 2019. And this is one where you're doing a task of reordering letters in alphabetical order. And this was an exciting paper because what they actually show is that in the prefrontal cortex, right, so way away from all of these very basic areas, that you can actually see a difference in the hemodynamic response in the superficial layers when there's being where the letters are being reorganized into alphabetical order, and an increased response in the lower layers with a response minus rest. So this was really very impressive, and also very encouraging that we could take laminar fMRI beyond early regions. So, of course, the next interesting thing is, can we do anything with connectivity? And just to maybe recap, functional connectivity is the underlying connectivity that we have when the brain is at rest. So it's what we often associate with resting state connectivity. And if we could do this at a laminar level, we could better understand structure-function relationships. You'd have a kind of baseline for the connectivity in the brain. You could generate a high-resolution connectome, and you would have a tool also ultimately for looking at disease progression. Effective connectivity is the influence that one region is exerting on another region when a task is being performed. And this is about the interactions of layers with other regions, or layer to layer within a region even. So these are kind of goals which are actually within reach. I hope that by the end of this talk I will have convinced you that that they are accessible. So at the base of all of this is a canonical microcircuit. So essentially, at the sort of level we're working on a lot in laminar fMRI, we consider a 3-layer rather than a 6-layer isocortex, and we have feed-forward connections projecting upwards into layer 4, and we have feedback connections projecting outside of layer 4. And you can either find both of these, or in some examples only one. So the Peter Koch experiment was quite likely a situation where there was a strong feedback signal into the lower layers when there was the surprise or the mismatch of seeing a triangle which didn't really exist. But armed with the canonical microcircuit, we can get information about the feed-forward and feedback relationships between different areas. And we can also look at the interactions between layers within an area. So again, John got there pretty early. Actually, this is a conference abstract from 2010, which I remember, and I've shamelessly copied it in here, which is showing the resting state interaction between human V1 and human HMT plus. Later, there was a development with vaso, showing very similar things here, shown over on the right. So you show a connectivity measure between the thalamus and the left B1, where the signal's coming in at the layer 4, where you would expect. But if you have the seed in HMT plus going into left B1, you're getting a double peak, which is consistent with feedback into the superficial and the inferior layers in V1. And this is just a control measurement here. And there's also, in the same paper from Renzo, very nice first connectome, where you can look at the connectivity between different regions. Again, here, V1 to V5. V5 is another name for HMT plus. And the interest is not in seeing layer-to-layer connectivity on the diagonal so much. It's always the interest to show things which are coming off the diagonal, showing an asymmetrical relationship between a higher and a lower region. And I think this is very interesting, but it maybe could be followed up more. I'm not also sure what the neuroscience community makes of these attempts and how interpretable they are. If we're looking at effective connectivity, you would probably have thought when you were looking at the chart of the canonical model that there's an awful lot of arrows flying around in there, right? So how can we imagine this? And I think it's quite illustrative to think that when you're doing normal bold fMRI, you're quite happy to have the concept of the general linear model, that everything is linear and additive. So given that different processes, different independent processes can be generating vasodilators and vasoconstrictors, then we can possibly see the net signals that we get as being the sum of the effects of all of these, right? Now, if, of course, these things have different functions, for example, there's the main effect of task, which is driving a certain response, and then there are feedback processes on top of that, then the feedback process may just be in the variance that we record, but we don't know unless we have a good regressor. So a lot of the things I will talk about in the last part of my talk ultimately rely on having some kind of independent signal acting as a regressor, which allows you to tease out that extra information. So we'll now look at effective connectivity in the context of a laminar fMRI study of language, where the language is very much in inverted commas because it's only firing words at people. So we won't go into enormous detail here, but it's a standard gradient echo bold 0.9 millimeter, and the interest is always between real words and pseudo words, and this is the contrast we will look at in multiple different ways here. So we look originally at a region of interest in the occipital temporal sulcus and its interaction with the rest of the brain. And the second part of this language bit will move further towards the prefrontal region. But this is essentially a region in the brain which is defined by having a larger activation at the macro level to pseudo words than to real words. And just to give you a feel for the data, these are the services. This is the data with the boundaries sketched in. This is the ROI in blue for one subject. And this is the layering white matter deep, middle, superficial, external. So this is the contrast real minus pseudo word. The whole region is defined already in the literature, in the language literature, by having a greater activation for pseudo words than for real words. And the first thing we found, which was kind of sticking out, was yes, it's true, but only when you average over all three layers. Actually, the deeper layer is showing the reverse contrast. So this was already something novel. When we looked at the interactions between the regions, we did psychophysiological interaction, which is a standard tool for cognitive neuroscientists looking at the interactions between regions. And what we found was a significant interaction between the deep and the middle region, which is consistent with the deep layer inhibiting the activation in the other layers when there's a real word compared to a pseudo word. So we're looking at the scenario where we think there is feedback from higher regions coming to the deep layer and inhibiting that response. Now, if we take the region of interest and look at the task independent connectivity, which would be a form of functional connectivity, then you can see that the middle bin, which is the region which would be getting input from lower regions, is very nicely showing up down with the early visual areas, whereas the deep bin is only linking up with some of the other language areas over here. So on the left side of the brain. If we go for an analysis of the interaction of the deep layer against the rest of the brain, so a layer to brain analysis, then we find that the deep bin targets the left lateralized language regions. And if we just moved up 1 millimeter, this disappeared. Right, so it's a fairly convincing evidence for an effective connectivity interaction in this task. One of the things is when you get these data sets, you can't stop reanalyzing them. So Daniel Charot, who was the prime mover on this, then of course we acquired nearly whole brain. And he started looking further into these data. So he picked out 3 regions which are active. Even though you're just reading the words, there is processing going on throughout the whole language system. And these 3 nodes are actually anatomically connected. So, and if we look, what he's basically done now is gone to BA44, which is a language region, and he's layered it, and he wants to look at the interaction between that and these 2 other regions. So this one is much more acoustic phonetic, interacting with node 2, which is in BA44, and this is much more, node 3 is much more articulation and motor cortex. So we also wanted to look at the interaction between these 2 regions. And essentially what we are expecting is a feed-forward relationship between node 1 and node 2, and a kind of feedback relationship between node 3 and node 2. And sorry, my slide change is somehow stuttering. And this again was done using the PPI, which is a standard technique, but it's just slightly adapted for laminar analysis. And basically, the result is quite convincing. Daniel went into more detail looking at long words and short words, but we don't need to go into that level of detail here. But if you look for the interaction with node 3, this is superficial greater than middle or deep greater than middle. You're seeing exactly this kind of interaction where the deep and the superficial layers are interacting strongest with node 3, whereas if you look at the interaction with node 2, superficial minus middle and deep minus middle are showing the reverse, that this is negative, so the interaction with the middle layer is strongest between node 1 and node 2. So we've succeeded here in identifying a feed forward and a feedback scenario deep in the brain. Now, that was a study in which we essentially used the, if you like, the signal from the other regions as the extra information to tease out the unique signal within the region we were looking at where we did the layer analysis. Another study which we did some years ago, and again has also produced a more recent publication, but I'll just show you the basic experiment today, is doing combined EEG and laminar specific fMRI. And here we want to do something different. We want to get an external regressor, and that's coming from the EEG signal. And we knew from previous work that the alpha and gamma signals, so to speak, the low frequency and high frequency signals had different representations in the bold. And also we knew from non-human primate experiments that the gamma is mainly coming from the supragranular layer in early visual, and the alpha from infragranular. So the aim was rather ambitious, and it cost an awful lot of time and effort, but the aim was to combine layer-specific fMRI with EEG to test this in humans. And there's a task where you look at inward moving rings, which generates actually a lot of gamma signal. So when you're looking at these kinds of plots here, you can see monkey V1, human MEG, human EG, and here the frequency range, so the gamma is here up at the 60 Hertz, and the alpha is down here in the low frequency range. When this work was done, with the analysis was split for the high frequency and the low frequency. So we basically perform this experiment in the scanner. We do not scan during the trial because the measurement of gamma is very sensitive, and we had an attention task embedded in this because sometimes they were expecting a speed change but it never happened, and sometimes they were not expecting a speed change and they saw exactly the same thing. So there are two contrasts embedded in this study. And again, it's just a standard gradient echo bold, 0.75 isotropic. We had to do it at 3 Tesla because we didn't have a safe EEG at 7 Tesla at that time. So if you look at the experiment going on, fixation, waiting for the motion, inward movement, speed change, recording, we can see the generation of the high frequency and the low frequency signal, which we recorded on the subjects in the scanner. And we can correlate the single trial EEG power estimates for all the frequencies with the bold signal from the layers in the cortex. So we're not interested in the main effect of task anymore. We've got independent regressors for the different frequency bands, which we can then use to start looking at the signal. And just to get the basic results out, we've got the change from baseline in the gamma, alpha, and beta. We've got the attention effect, which is weaker because it's an attention effect and it's also a smaller number of trials. And if you look at the depth dependency of the fMRI attention effect, averaging over all visual regions, or V1, V2, and V3, it's clearly there, broadly present. And for us back then, when we got this result, we were truly amazed, because of course back in those days people were not so believing in laminar fMRI as we are perhaps now. But when we looked at combining across all of the regions, we look at the high frequency, then the gamma is only correlating, the gamma variant is only correlating with bold signal in the superficial layers, whereas the alpha and the beta is going through. And you can see more or less the same pattern in V1, V2, and V3 independently. And if you look at the attention effects, then this is the combined attention effect. Again, the gamma is in red. This is coming up here in total in the superficial layers. What's interesting now is that the alpha is only showing up in the superficial and not in the deep regions, whereas the gamma is showing up in the beta, I beg your pardon, is showing up in the deep regions. So this is also speaking to the analysis of these effects not being dominated by the leakage of the signal from the lower layers to the higher layers. So amazingly, perhaps I'm going to make myself popular here, because I understood from John that I'm the person between you and your lunch break. We've come close to the conclusion of this talk. So the lessons learned over all these years. Well, in general, I think on the acquisition and analysis side, there are many ways you can do things wrong in this, but there's no single way of getting it right. So Renzo has a wonderful talk on pulse sequence wars, which probably you'll see at some stage. Most sequences will produce answers which are hopefully very similar. At the end of the day, we're all looking at functional hyperemia. So if you do your experiment well, you should get a good result out of it. When you're designing your experiments, work backwards from the scientific question you're asking to the expected activation pattern, to the analysis technique, and then to the acquisition protocol would be my advice. The standard considerations are always going to be there, right? So we should always be asking the same questions: temporal spatial resolution, volume coverage, sensitivity, specificity. I pay particular attention to whether you're in a physiological or a thermal noise dominated regime, because there are techniques for removing or reducing physiological noise. There are also now very cool techniques like Nordic for reducing thermal noise, all of which are going to have an impact. So the topical answers to these questions will definitely evolve over time. But I think the questions you should be asking are more or less the same always, right? I think we would have been asking the same questions 10 years ago. We would have probably just given different answers than we are giving now. And then maybe you can try that and see if the faculty agrees with me over lunch. And on the optimistic note, I've been involved in this field for a very long time. And we've gone from skepticism to euphoria, I think. People originally said, are you insane? Everybody knows that you can't do anything better than 3 millimeters with the functional hyperemia. And now we see that we're getting real applications which can hopefully answer a lot of questions in neuroscience. I think it's also now a very pleasant community to work in. It's got a great spirit, a lot of supportive people who are willing to share all sorts of code and expertise. I won't stop without acknowledging people who actually do all the work. I would particularly point out Peter Koopmans, whose PhD was the pioneering PhD on laminar fMRI in our group. Irati and Tim, who did a lot of analysis on the simulations and the spatial GLM. René Scheringer, who was really the man who did the EEG fMRI and cost an enormous amount of work to get those results. And similarly, Daniel Charot, who's done all the language work. So on that note, thank you for your attention. I hope you found it informative, and I'd be happy to take questions.